Hello, my name is Joshua Mugabi. I welcome you to Genes. Today, our lesson objectives will include the genes. We shall look at the loci, alleles, mutation, genome, Human Genome Project, HGP, and lastly we shall look at genetic comparison. You are welcome. Please do not forget to subscribe. Let's look at the genes. We start with the genes. Deoxyribonucleic acid, which is DNA, functions as a genetic blueprint for living organisms. DNA is the genetic print of living organisms. It means that it is the one that encodes for the proteins. It is the one that makes up the genes, which encodes for the proteins. It encodes proteins to carry out cellular processes needed to maintain life. A gene encodes proteins to carry out cellular processes needed to maintain life. So if you look here, we have our gene A. And gene A, this one is on a DNA chromosome, and that gene A encodes for protein A. Then we have another gene on the same chromosome. There is also gene B. This gene B is situated at this place, which we call a locus, and that gene encodes for protein B. So we are saying that genes, they encode proteins that carry out cellular processes that are needed to maintain life. Specific sequences of DNA that codes for a particular protein is called a DNA. A specific sequence of DNA that codes or encodes for a particular protein is called a gene. So a gene is a specific sequence of DNA that encodes for a particular protein is what we call a gene. Now this protein here, protein A, is encoded by a sequence of DNA. A sequence of DNA, that sequence there. It's the one that encodes this protein. So that specific sequence of DNA, which is this one here, is what we refer to as a gene. It is referred to as a gene. When you look at this side on part B, on the same chromosome, there is this sequence here. That sequence there of DNA, it encodes for this protein. And that sequence of DNA that encodes for this protein is the one that we are referring to as a gene. So it is an heritable factor. It is an heritable factor that influences a specific characteristic or trait. It is a character or a factor that can be inherited. It can be passed on to the offspring, and it influences a specific trait. It influences a specific trait or a specific character. That is a gene. Every living organism inherits a blueprint for life from its parent. Every living organism inherits a blueprint for life from its parents. That is the sequence of DNA. The DNA is the blueprint that is inherited from the parents. And the sequence of DNA that codes for particular proteins is what we are referring to as a gene. So a gene is still the blueprint. A gene is made up of sequence of DNA that encodes for particular proteins that carry out particular life processes or cellular processes. A gene is a heritable factor, that is a gene, and it consists of a length of DNA and it influences a specific characteristics. So we have a gene which is a heritable character and it is made up of a particular length of DNA. It is made up of a particular length of DNA which we are calling a sequence of DNA, and that particular gene influences a particular character. It influences a particular character. Let's talk about the gene loci. The gene or the specific position of a gene on a chromosome is called a locus. In plural, they are loci or loci. The specific position of a gene on a chromosome is what we refer to as a locus or loci. That is the specific position where the gene is located. In eukaryotic genes, loci are identified according to a number of differentiating features. That is for eukaryotic gene loci. They are identified according to the number of differentiating features. 1. The chromosome number. The chromosome number. Is it chromosome 7, chromosome 1, chromosome 2, chromosome 3? We can use that one. Secondly, the arm. It can either be the P arm, which is the shortest, or it can be the Q arm. Thirdly, the region and the bands. We can use the region and the bands. Region 15.2 or band 31.3. Region and band 31.3. So when we identify the loci or the locus of a particular gene, we can identify it using the number, chromosome number. It can be chromosome number, for example, you can say chromosome 7 and on P arm, which is the short arm, and the region and the band is 15.2. So there we would have identified the gene's locus. We use the chromosome number, we can use the arm, and then we use the region and the band. A gene occupies a specific position on a chromosome. Let's look now on the allele. The allele. Allele are different possible variables or variations of a gene. We can say they are the alternative form of a given gene. What we call alleles, these are the different possible variations of a gene. Or we can refer to them as the alternative forms of the gene. The relationship can be described as follows. A gene encodes a genocharacteristic. A gene encodes a genocharacteristic. Like for example, when you say the eye color is a genocharacteristics. A genocharacteristic, and that one is encoded by a gene. Whereas the allele, these ones encode for a specific alternative. It encodes for a specific alternative. For example, blue eyes. Blue eyes. Now, for example, if we look, we say eye color. Eye color. They are what we are talking about. We are talking about the gene is eye color. But when we go deep further and they specify and say blue color, now we are talking about an allele. It is the gene is the eye color, the allele is the blue color, or we can say brown eye. Brown eye. Eye color is the gene, and the brown eye now becomes the allele. So the alternative is brown and blue. Those are alternative, but all of them are eye color. So that's why we are saying that a gene, for it encodes the genocharacteristic, which is eye color. But the alternatives of eye color, the eye color can be blue or it can be brown. Now that one is what we call the alleles, alternative forms of a gene. So alleles differ from each other by one or few bases. The alleles, they are going to differ from each other by just one or a few bases. You have allele for brown color or allele for blue color. They have similar bases, but they just differ by just a few bases. All of them, they are eye color. Gene, it's a gene, the same gene of eye color, but these ones are the alternative form. The allele for brown color, and then another one is allele for blue color. But all of them, they are eye color. Eye color, which forms the gene, and then the different colors, they form the alternative form of the gene. Therefore, those ones now we refer to them as alleles. So, majority of the gene sequence is conserved. For example, if you look at the eye color, brown eye color, and then you look at the blue eye color, majority of the gene sequence is conserved, is the same, but they are just going to differ from each other by just one or a few bases. They will differ from each other by one or a few bases, but the majority of the gene sequence is conserved in both of them. So the various specific forms of a gene are what we call alleles, and these ones may differ from each other by one or only a few bases. Let's talk about mutation. We have a gene mutation This one is a change in the nucleotide sequence of a gene. It is a change in the nucleotide sequence of a gene. Okay? For example, we looked at a gene as a sequence of DNA. And DNA is made up of nucleotides. So a specific change in a particular sequence, nucleotide, of a gene is what we refer to as gene mutation. For example, here, if you look, we have thymine, adenine, cytosine, thymine, guanine, cytosine, and guanine. Now, when we come this side, we have thymine, adenine, and here we are supposed to have another cytosine, but now there is mutation which has taken place here. Instead of having cytosine, now we are having thymine. So that one means that there has been a change in the nucleotide sequence of that particular gene. So that change in the nucleotide sequence of a gene is what we refer to as mutation. Changing the gene sequence may change the function of the encoded protein. So here, because we have this gene sequence which is encoding a particular protein, and that particular protein has a specific function. So when there is mutation, if mutation takes place, then that means we are going to change the protein which is going to be formed. And that one may change the function of that particular protein which is formed. May not have the original function like the gene which is going to be encoded by the sequence which has got no mutation. Gene mutation can either be, for example, it can be somatic. It can be somatic, and the one for somatic, this one occurs in body cells, and it cannot be inherited. It cannot be inherited, but it may give rise to cancers. So that one is what we are calling somatic mutation. The type of gene mutation which occurs in body cells cannot be inherited, but it can cause cancers. Then we have another gene mutation that can take place in germline. This one occurs in sex cells, or what we refer to as the gametes, and it can be passed on to the offspring because it is taking place in the gamete cells. New alleles are formed by mutation. So when we do have mutation taking place, it is going to form other alternative forms of a gene. Those ones are the ones now that we are referring to as the allele. So a new allele may be formed because of mutation taking place. Mutagens. A mutagen is an agent that changes or mutates the genetic material of an organism. Something that changes the genetic sequence, something that changes the genetic material of an organism is what we refer to as a mutagen. The mutagens may be physical, they can be chemical, they can be biological in origin. For example, if it is physical, like here we can see, we have ultraviolet light and X-rays. Those are physical origin. Then we have the chemical, we have, like, for example, carcinogens, we have the processed foods, chemicals, we have the cleaning products. All those ones can have or can be called the mutagens, chemical mutagens. Then the biological one, we may have the viruses, and then we have the bacteria. So those ones that can cause mutation, those agents are either physical, like X-rays and ultraviolet light. They can either be chemicals, especially those ones that are found in cigarettes, chemicals found in processed foods, chemicals found in detergents. They can also cause mutation. And then we have the biological. Those ones are from viruses and bacteria. Let's look at the types of mutation. We have what we are referring to as point mutation. This one involves the modification of a single nucleotide in a sequence. Point mutation, it involves the modification of a single nucleotide within the sequence. That one is a point mutation. And this one can either involve substitution, it can be insertion, or it can be deletion. And it is modifying just a single nucleotide. But it can involve substitution, it can involve insertion, it can involve deletion. Now, substitution mutation can either cause what we call silent mutation. In a silent mutation, no change to the polypeptide sequence. There is no change to the polypeptide sequence, as we shall see later on. This is due to the genelens. It can be a missense mutation, whereby a single amino acid is hanged. Here, a single amino acid is hanged in a polypeptide. Still, that one is a substitution mutation. Nonsense, it can be a nonsense mutation. In a nonsense mutation, it's where a stop codon is created and therefore truncating the polypeptide, cutting off the polypeptide. We create a stop codon. That type of mutation is what we are referring to as nonsense mutation, and it is a substitution mutation. Then it can either be insertions or deletion, and these ones can cause what we are referring to as frame shift mutation. In frame shift mutation, it changes the reading frame. It changes the reading frame of the sequences. Mutation examples. We have an example of sickle cells. Now, sickle cell anemia is caused by base substitution in hemoglobin. Sickle cell anemia is caused by base substitution in hemoglobin. Just a base is substituted in hemoglobin sequence. And when it is substituted, it ends up forming what we call sickle cell anemia. For example, here, if you look at the DNA antisense, we have the normal hemoglobin. It has cytosine, thiamine, and cytosine. So when you look at messenger RNA will have guanine, adenine, and then guanine. So this one will encode for an amino acid which is glutamine. Now, if there is mutation taking place, and that's one resulted from base substitution, instead of having thiamine, we substitute it with adenine. Thiamine is substituted with adenine. So now we shall have our bases as cytosine, adenine, and the cytosine. Now what will be read encoded on messenger RNA will be guanine, uracil, and the guanine. So now the protein which is going to be encoded for is going to be valine. This is different from glutamine. So we are going to form a different amino acid. Now this one, because of this, now we are going to move from a normal hemoglobin. Now we are going to form what we call a sickle cell variant because of this amino acid which we have formed here. due to the change, just the substitution, because of the substitution of a single base. So the 6th codon in sense strand, for example, if you look, it is guanine, adenine, guanine. Now it gives us guanine, thiamine, guanine. And this one is now going to change the reading. And now we shall form amino acids. Instead of forming glutamine, we shall form valine. That is a change. And when that one changes, of course, it will now move from being a normal hemoglobin to form a sickle cell variant. Causes of sickle cell anemia includes the base, substitution, mutation, and this one changes the messenger RNA transcript. It is going to change the messenger RNA transcript. This is the normal messenger RNA transcript, but now this is the changed messenger RNA transcript. Because of the base substitution, the bases have been interchanged. Instead of having adenine, we are having uracil. So that one is the one that causes our sickle cell anemia. Please don't forget to subscribe. Let's look at mutation sequence, which is anemia mutation sequence. Now, the amino acid change. Instead of having GLU, we are now having VAL. And this alters the structure of the beta chain. And when the structure of the beta chain is changed, this is going to make hemoglobin to form insoluble strands. Because of the change or the altering of the structure of the beta chain. Now, the insoluble strands, these ones are going to change the shape of the red blood cells. And they are going to cause it to form what we call a sickle shape instead of forming a biconcave shape. So we are seeing, because we have changed the amino acid from GLU, which forms the normal shape, which is biconcave shape. Because of that change of a single base amino acid from GLU to valine, now valine is going to form what we are calling a sickle cell shape. The red blood cells cannot carry oxygen effectively and are destroyed at a higher rate, leading to anemia. Now, if you look at this red blood cell, this one is biconcave and it carries a lot of oxygen. This one is sickle-celled. It is sickle-celled and it cannot carry enough oxygen. And because it cannot carry enough oxygen, they are going to be destroyed at a higher rate. And when they are destroyed at a higher rate, this will result in a condition which we call anemia. So sickle cells are also likely to form damaging clots. They are likely to form damaging clots in the blood vessels. Causes of sickle cell anemia, we have said that includes the changing of the sequence in the polypeptide of hemoglobin. If we change the sequence of the polypeptide in hemoglobin, we are going to cause what we are calling sickle cell anemia. So let us look at the genome. When we talk of a genome, this one describes the totality of the genetic information in a cell. Total genetic information in a cell or an organism is what we refer to as the genome. The totality of genetic information in a cell is what we are referring to as an genome. For example, here, if we give our genetic information, we have genetics, then we form the genes, then we form what we call the genome. For example, in computers, if it is technology relationship, we have the binaries, then they will form programs, and programs will come and form the computer. So it includes all the genes and the non-coding sequences. Of course, there are those sequences which are used for coding the genes, and then there are other sequences which are non-coding. So all of them, when you add them together, they are the ones that form what we call a genome. So genome is the whole of the genetic information of an organism. All of the genetic information of an organism is what we refer to as the genome. Human genome. Human genome. So we're looking now at the human genome. The Human Genome Project, HGP, was an international cooperative venture which was established to sequence the entirety of human genome. It is a project that was established to sequence the entirety of the human genome. It was found that humans share a majority of their sequences, with only short nucleotide differences contributing to genetic diversity seen in mankind. So, all humans, they share the majority of their sequence, with only short nucleotide differences. There are very few short, simple nucleotide differences that are contributing to the genetic diversity that we see in mankind. But mainly, we share the majority of the sequence. Humans, they have roughly 3 billion bare pieces of DNA in their genome. That is the total genome. And the human genome typically consists of 46 chromosomes. That one is for all of us. And the genome contains roughly 20,000 protein coding genes. So that is the human genome. It is roughly 3 billion base pair of DNA in their genome. And the genome typically consists of 46 chromosomes. And it contains roughly 20,000 protein coding genes. The entire base sequence of human genome was sequenced in the Human Genome Project. Let's look at genetic comparison. Separate species, they have distinctive genetic patterns that differ in a number of ways. So if we have different species, they have distinct genetic patterns, and these ones are differing in a number of ways. And this includes the genome size, the number of chromosomes, and the number of genes. So different species may have different genome size, they may have different number of chromosomes, and they may have different number of genes. There is no clear correlation between these factors. However, certain traits do exist. Viruses and prokaryotes. Viruses and prokaryotes. Prokaryotes, those are organisms which don't have a nuclear membrane. They tend to have smaller genomes and fewer genes. Those are viruses and the prokaryotes. Then the eukaryotes. Eukaryotes, these are organisms that have got a nuclear membrane or cell membrane bound organelles. They tend to have a huge variation both in the genome size and the gene numbers. They have a very huge variation in their genome size and the gene number. Those are eukaryotes. The overall, there is no consistent or reliable pattern between these genetic factors and relative complexity of any given organism. No consistent or reliable pattern between the genetic factors and related complexity of any given organism. Please do not forget to subscribe. Comparison. We want to look at the genome size. Now, the genome size can vary greatly and is not a valid indicator of genetic complexity. The genome size can vary greatly and is not a valid indicator of genetic complexity. Now here, if we look at the different species, their genome size, for example, we have the viruses. Their genome size is 170,000 base pairs. When you look at the bacteria, this one is Escherichia coli. It has 4.6 million base pairs. When you look at the fruit fly has 130 million base pairs. If you look at human, Homo sapien, we have 3.2 billion base pairs. And then if you look at the canopy plant, Pallis japonica, it has 150 billion base pair. That is the genome size. But they have told us that the genome size can vary greatly and is not a valid indicator of genetic complexity. So if we are looking at it, that the more you have, the more you become more complex. Then we would expect the canopy plant to be more complex because it has got 150 billion base pairs compared to Homo sapiens with 3.2 billion base pairs. So they are saying that that one does not validate or it does not indicate the genetic complexity. Humans have a genome size of roughly 3.2 billion base pairs. Now we want to comparison on the chromosome number. The chromosome number is the characteristic features of a member of a given species. Chromosome number is a characteristic feature of members of a given species. So we have the roundworms. This is a species of Parascaris equiram. They have 4 chromosomes. Then we have rice, which is a species of Rhiza sativa. They have 24 chromosomes. We have humans in the species of Homo sapien. They have 46. We have the chimpanzee in the species of Pantroglodytes. They have 48. And then we have the dogs, which are species of Canis familiaris. They have 78 chromosomes. Humans have a diploid chromosome number, which is of 46 chromosomes, and the N is 23. That is the pairs. The pairs are 23. and the diploid number is 46. Comparison in gene number. Estimation of gene number varies significantly depending on the prediction method. For example, the bacteria, the genome size is approximately 4,200. Chicken, it's approximately 17,000. Humans, it's approximately 21,000. We have water flea, it's approximately 31,000. And rice is approximately 38,000. Humans are estimated to have between 19,000 to 22,000 protein coding genes. That is what is estimated for humans. Genetic databases. There are a number of online databases that allow for comparison of genetic characteristics, including bases and polypeptide sequences, lossy positions, and many others. Gene banks contains the collection of DNA and polypeptide sequences. Cluster Omega is an alignment program for comparing different sequences. So hello my friend, thank you very much for participating, and today we have looked at the genes, we have looked at the loci, we have talked about the alleles, we have talked about mutation, we have seen the genome, we have looked at Human Genome Project, and lastly we looked at the genetic comparison. Thank you. And please do not forget to subscribe. Thank you.